Okay, welcome back. This is part four. We're going to really get into it now. We're going to uh, get into some of the things you should already know about. Just passive transport. This is really just general diffusion. But there's one thing I wanted to point out. You don't really see it in this diagram, but there's something going on that is important. What we have is Brownian movement. Now, of course, that is directly proportional to the temperature. The higher the temperature, the more of that Brownian shaking and a Brownian movement, and the molecules bump into each other. And that really dictates the rate of diffusion that goes on. So, if you put this drop of food coloring in, the time it takes for it to even out through the solution is really dependent on the temperature and if there's any shaking of the bowl or whatever. But that's usually the overall writing characteristic. So the phospholipid bilayers of the membranes really offer some selectivity just in the fact that you have these hydrophobic regions and hydrophobic in this area and the hydrophilic regions on the outside. And there is some pressure, although individually weak but collectively very strong. Uh, Various things are excluded or are allowed in. So, in simple diffusion, small molecules like water and oxygen and various things can diffuse across, but larger particles require some sort of facilitation. And this is usually something that's been, of course, genetically encoded and then goes through the process of the DNA to the RNA to a protein, and that becomes, if it's a gated process or something, it's some protein that's embedded in the membrane. So this region of the folding of the protein would be hydrophobic, and then these regions out here would be hydrophilic, and it sort of allows it to kind of float in that orientation. And then the channel will have certain charges that are consistent with whatever it needs to facilitate across the membrane. It would be compatible, size, charge, and shape compatible, so it'll allow things to come in. So this doesn't require energy, but I always say it did require energy at the initial stage to have the protein, of course, made in the Golgi in the endoplasmic reticulum and then shipped to the Golgi apparatus. So that's pretty much where we are on that. So for proper functioning of cells, we must acquire food and other necessary materials, and we have to get that from the outside, so we have to bring it in. And we also have to release certain waste materials and the like. And so in passive transport, which is really just simple and facilitated diffusion, as I went over, and this takes place in the molecules as they move down the concentration gradient. So we're going from a higher concentration to a lower, and that's kind of how it works. Now, osmosis. This is different. I don't know how many times you've been exposed to osmosis, but I mean, in terms of school, your body's constantly undergoing osmosis. Your kidneys are filtering and all these sorts of things. So tonicity refers to the concentration of solutes. Now, solute is what's floating in solution, and we have solutes that are inside and outside. Now, keep in mind that solutes, for whatever that substance is taking up space, i.e., uh, it's negating water in that space. So it really comes down to how much water we're talking about from one side of the membrane to the other. So if we're displacing water with that solute, then we have less water on one side of the membrane, if that makes sense to you. So what happens ultimately is gravity. Gravity will make what they call osmotic pressure. That pressure between the differences between the two sides of that membrane will equalize based on the gravity effects on water, and so that you'll get the same. Now the levels may be different, but it will be accurate as it relates to the displacement of the water. So that's where we're at on that. So there are some aspects of osmosis when we talk about the net movement of water. And so isotonic is the concentration of solutes outside the cell is equal to the concentration inside. Hypotonic is the concentration of solutes outside the cell is lower than the constant. In other words, we have more displacement of water inside the cell than the outside, and we refer to that as hypotonic because water concentration will be lower. Hypertonic, the concentration of solutes outside the cell is higher than the concentration inside, so there's more water displacement outside than there is inside. So hypertonic, the concentration of solutes outside the cell is higher, and so the amount of water will be lower in the outside of the cell. I hope that makes sense to you, because we're going to kind of look at it again, but in a different way. So isotonic, we have sort of an equal balance of water, so the osmotic pressure is the same. Hypotonic, the solute concentration is lower in the extracellular, so it's lower and we have more here. So what's going to happen is that we're going to try to equalize the amount of water, and the water is actually going to go in to the cell, and we call this hypotonic solution. And what usually causes is cells to burst, because you're adding more water than the capacity of the cell can handle. And for us, we have that issue with, we have 0.95% salt concentration in our blood, and that's one of the measures of that we can use for osmosis, because if we now introduce this regular, let's say, distilled water, then there is no solute, there's no floating salt in the water that we're adding. And so the red blood cells, of course, have a certain amount of salt inside the cell, and if it's water is going to move into the cell to try to equalize that water pressure, because the salt is displacing inside the cell, and so as a result of the cell expands and will explode. And that's what we see. Hypertonic, this is the solute concentrations higher in the extracellular, as we discussed, so the water is going to move out of the cell and it becomes dehydrated, or it shrivels up, and that happens when the concentration outside is higher. So I had an incident once where I was bitten by a cat. I was working for a vet, and right through my thumb and all this, and so they had to extract the cat from my hand, but he bit through my nail and all sorts of things, so I lost some blood and all these things. So it was during a shift change, and for some reason I got ringers and they didn't add the salt to it, and as a result I started to feel really bad. And of course, when you add ringers without the salt concentration, it's sort of like adding fluids, but the osmotic pressure was wrong. So I had pure water going in, bursting my red blood cells. So the doc acknowledged, saw this, and immediately corrected the situation. But I passed out as a result, and anyhow, that wasn't fun. So I'm a little reluctant to go into hospitals for procedures during a shift change. Just me, I don't know, whatever. So the solute size informs selective permeability. So we have a membrane that is selected to a particular size of particulate. So each of those ball sizes displace a certain amount of water. The smaller ones can pass, so they'll equalize. That's not the issue. And a lot of times problems or situations that you may come across, once these are permeable to that membrane, you don't even worry about it, because the amount of water is going to be the same, but it's the larger particulate matter on one side of the membrane than the other, so this is going to displace more water. And so osmotic pressure then is you're going to have water trying to move into this to separate those, to equalize that osmotic pressure. So if you think about it that way, I think life will be much better for you. So dehydrated celery contains many dissolved molecules, solutes relative to water molecules. And so there's water and the dissolved molecules. When placed in distilled water, contains fewer dissolved molecules than the celery does. The water molecules diffuse into the celery, equalizing water concentration inside and out, and the celery becomes turgid or more crisp. In other words, it's filling up with water to try to equalize. And plants tend to favor that a little bit because, of course, the whole deal, as we're going to talk about, is they use water for their energy. In other words, water is cracked during photosynthesis into hydrogen, and that charges batteries, and that spins ATP synthase, which we'll talk about in the energy chapter. But I don't mind trying to get you warmed up to that as we get to that topic. Then you're going to say, ho hum, I already know about that, which is good. Why distill water? Because it has very few dissolved molecules in it. Well, it shouldn't have any, and it is a hypotonic solution. So the cells in the celery stalk, on the other hand, contain many dissolved molecules such as salt. Because the dissolved molecules can't easily pass, the water molecules move into the celery, moving the pool. So what they're not saying is what's happening is that you're getting an equalization of the osmotic pressure to equalize the displacement of the sucrose molecules, and you'll get water molecules moving in as a result. And so you'll see this in a minute. When placed in salt water, would your celery regain its crispness if you place it in salt water? No, because the hypertonic solution, there are more dissolved molecules outside the celery cell. So what little water remains in the cell actually moves out through osmosis. So that's best demonstrated here, as you can see. What's going to happen is these are displacing the water, and you have more water space between these molecules. So what's going to happen is this gravity is going to act on this, and so these are displacing the amount of water. But if you were to look at it in terms of the weight, the sheer weight of the water, they're equal. Even though it's displaced and it's much higher on this side of the beaker, it's due to the fact that now the water pressure between the two sides are the same. And that's due to the displacement of the water by the larger sphere. And you can see, obviously, there's more water on this side than there is this side. So it's going to move over until that is equal, and always finds the perfect equilibrium there, just based on gravity and osmotic pressure of the water. So here's just another way of showing the same thing. And we have more of these spheres on this side than here, so we actually have more water over here. So when somebody asks you what's the net movement of the water, you can see there's more water on this side. It's going to move towards the area where it's not as much, and as a result, this level is going to go down and this level goes up until the osmotic pressure is equal between the two sides. And if you think of it that way, then you can survive any question about osmosis. And again, here's normal cells. Saline should be 0.95% salt, and it's normal, so it's isomatic solution. But if you have hyperosmotic red blood cells, what ultimately happens is you have more water on the outside than the inside, and so water is more water on the inside than the outside because you got hyperosmotic. So water is going to move out and it's going to cause the red blood cell to shrivel. And then, of course, if you have a higher concentration of water on the outside than the inside of the red blood cell, then it's going to move in and it will burst. And so that's not a good situation. It has to be corrected. So diffusion of water across the membrane is a special type of passive transport called osmosis. Water moves from an area of lower concentration to an area higher concentration because what you're doing with the driving forces, and the way I want you to look at these types of situations, is the net movement of the water and osmotic pressure, which is the effects of gravity on the water. So it's all about the displacement of that water being under the effects of gravity. So that's one way to look at it. So that would ask an interesting question. So what happens to the astronauts in space when they don't have the effects of gravity? Think about that. So we also now have to move things sometimes that isn't it's not an easy thing, and so we actually have to have move materials across the gradient to a higher concentration because we need it. And so we use energy to do that in the form of chemical bond energy provided by the universal currency of the cell. It's called ATP, adenosine triphosphate. We've already talked about that, but it doesn't hurt to just mind you. So ATP will lose one of the phosphates. The chemical bond energy associated with that phosphate bond is about 32 kilocalories per mole, which means that it'll raise the temperature of water because of that chemical bond energy. Well, it either does it by transforms in heat, or it transforms the energy. It was never lost or created. We're going to talk about that. It just transforms, and so that energy is transformed into the changing of the shape of this carrier molecule, and it will move things. So it'll actually bring it in and then pinch, and then it pushes it inside, whatever, in this case it's hydrogen ions or whatever. And so active transport will do that. Now there's something interesting about this. Because you're getting a higher concentration in the region because you're forcing it, sometimes in secondary active transport, the transport protein moves one molecule against its concentration gradient, powered by the simultaneously flow of another molecule down its concentration gradient. So sometimes when you pay for the movement of ions, you'll actually get a secondary active transport. In other words, riding on the fact that you've got higher concentrations that you've had to spend energy money for, and so we call that the secondary transport, and that's due to the overflow or concentration effects down to another. And it's just nothing is wasted. Everything is very efficient. So active transport is that we're paying to play, really, essentially. And the analogy I use for that, of course, you got to put in a money card or your cash or something to open the door and allows you in. That's active transport in a way, in an analogy. So when you pay, in other words, you put in the coin or the prepaid card into the machine, it opens up and you're allowed to transfer in the protein. The real, where the analogy kind of breaks down, is the protein changes its shape and literally pushes whatever it is that they want to move. Now, I've been in some ones in Japan. You push the thing and it kind of does spank you on the backside, but that's not really a good analogy. But anyhow, there are two distinct types: primary and secondary. We've already talked about the primary, which is where we actually doing the work of changing the shape or conformation of that protein in the membrane due to the chemical bond energy of ATP, and then you increase that concentration across that particular membrane. Secondary transport mechanisms will start to work, and that's called secondary. And I'll go over it one more time. So we have two distinct types. We have active transport, which uses ATP. The second active transport actually has to utilize the primary, but there's because of the higher concentration, there's some additive benefits for that. So not bad. So many transporter proteins use an indirect method of fueling their activities, and so the idea behind this, yeah, so it goes to the higher. So you can see when we get more across that membrane, uh, the more of that particular substrate has been moved across, and the secondary of course takes advantage of it. But the primary, the only thing I really want you to know, you can watch the YouTube, you probably didn't hear it as well. I'm still working out some of the glitches here, but you can use that YouTube link right there and cut and paste it right into your Google Chrome browser, and you could watch the video. It's a little bit more detailed than I really wanted to go into, but it's nice that you can see some a video on that. Okay. Whoops. So in active transport, moving molecules across the membrane requires energy. That's why we call it active. Active transport is necessary if the molecules to be moved are very large or being moved against a concentration gradient. And stop that. Proteins embedded in the plasma membrane act like motorized revolving doors. And so when it does that, you increase whatever it's moving across in concentration. It may have an effect in another transport system, just the fact that we have a higher concentration, which now causes that to move certain substances across the membrane. And that's the secondary part that doesn't require technically ATP like the primary does, but it would not happen unless the primary was working. If it doesn't work, then the secondary shuts down as well. So they're linked, of course. So two terms. A lot of this is vocabulary, and that's why I assigned some of the vocabulary for you to define, because technically in biology that's what you should be studying. No one really talks about it, but vocabulary is important, and when you get test questions and the like, a lot of it is just knowing and identifying vocabulary words. So if you know that up front, it sure makes studying a whole lot easier. If you look at the vocabulary words. Now I also supplemented somewhere, it's in the additional resources on Blackboard, but I've given you some Latin terms that reference some biology, and that way you can see the roots of some of the words. Instead of just memorizing, you can see where the Latin terms come from, and I like to view that as systematically learning, because you can see the roots of the words come from the Latin, which a lot of the English or German or Russian or other languages kind of borrowed or derived some of the language. And it's just nice to know, having those terms under your belt that are in Latin. It sure makes learning a lot easier, and so I always try to encourage that. So endo, inside the cell, and osis means the process of. Usually each part of that word is divided by this connecting vowel, which is an O. It doesn't always have to be an O, but endo is within, cyto or site is cell, and cis or osis is the process of. So it's the process of bringing something into the cell. And that way I'm systematically learning the word, so I don't have to memorize it. Systematically, then you can kind of figure it out on your own. And that's a better way to do it in the long term. So engulfing large particles with plasma membranes. So there's three types of this bringing things in. We can bring in solid forms, liquid forms, or specific types of forms, and we have names for these things. And so phagocytosis, phago means eat. Cyto is cell in the process of eating, the cell that has a process of eating. Pinocytosis, pino means drink in Latin, and you can think of it as a pint of beer or something like that. Pinocytosis, so it's a process that cells drink. And then we have receptor mediated endocytosis, which has a lock and key type of thing. And then of course what goes in goes out. Exo, exit signs, in Latin is exit, E-X-I-T. He, she, or it moves out, and so exo means out the cell, so it's a process of the cell moving things out, and that transports large molecules out. Not a big deal, but I just wanted to kind of show you the systematic way I try to learn these things as I was going through school myself, which, but be honest with you, I'm always learning. And I'm looking into new things. I can't even keep up in my own field anymore. There's just so much new stuff. So phagocytosis, phago, eat. So it's the cell that eats. So here's a large particle. So it invaginates it with that phospholipid bilayer, and now you have a vesicle surrounded by that phospholipid bilayer. That's how cells go through that process of eating. Now we also, you can see the word phago, eat, and pino, drink, and so the cytosis part is identical between the two. So it's a process that the cell drinks or the process that the cell eats. And it could be a large particle solid, or it could be a liquid, and that would be drinking, pinocytosis. Receptor mediated is that you have some sort of receptor and the outer side. So wherever there's a positive charge here, it's negative over here. And if it has a certain shape, it would be the opposite of that shape. So we have a kind of concord in, and then you have the circle-like shape, and it would fit inside. So it's like a key in a lock, and it fits in there. And then the cell recognizes that as sort of a switch, where it now starts, what they say, invaginates it inside, and now we have a vesicle like the other, but it's secured by specifically by the receptor, so we know exactly what we're trapping. And cholesterol can be used this way as an example. That's the way our cells kind of move that. The best analogy I have for that is on an aircraft carrier. We move planes above and below deck, and it goes through a series of, you can see there's above the deck, we see the plane up there, and so they move the deck down so they can store it underneath. And so somebody had to push a button, and that is sort of what's going on with the receptor, is you sort of push the button. So when the lock and key fits, it causes a shape change, and it eventually moves it in. And here you've had to trigger the elevator mechanism somehow to move the aircraft in there. Boy, that looks like a mean aircraft right there. Look at the teeth and all that. It's pretty wild. And anyhow, so the recognizes and binds a specific type of molecule, and so that's the key, and it's the lock and key, and so in it goes, and that's the receptor type. So we have those three different types. Now I'll give you a sort of an applied sense of this. An example of a receptor mediated endocytosis is cholesterol, which is as you get older, everything happens. Your hair falls off, your ears grow like Dumbo. I could go on and on, but I won't. But the other thing is the management of cholesterol tends to be a little bit harder because of less testosterone for men and that sort of thing. So most cholesterol that circulates in the bloodstream is in the form of low density, or LDL, lipoproteins. Each LDL is a cholesterol globule coated with phospholipids, and so it's the HDLs and LDLs are the kind of the two that we want to keep a balance of. And when the cholesterol levels exceed the LDL receptors, the capacity for binding it and removing it becomes reduced or blocked, and that becomes a problem, because then we have all these things floating around in our vascular system, and we have a healthy artery, and now we have, instead of this nice big opening for blood to transfer, it's all you can kind of think of pulmonary disease like this is sort of like a scab that doesn't heal, and it blocks the flow of blood. And I'll have a more technical view of this. You can see HDL, endothelial no production. It's kind of tied in. Here is the blockage occurring, and you see a buildup of this material, and we have sort of a scab that's not healing, and then it gets worse and worse because it's trapping. It becomes sticky. All sorts of bad things happen, and so too much circulating cholesterol in LDL molecules leads to cardiovascular disease. You want to have the good cholesterols around there to kind of help mop up some of that stuff. But here's the deal. We talked about trans fats. Nothing in this diagram deals with trans fats. It's either it's a fatty tissue that our system can either break down or not and convert. And trans fats just stick all sorts through these sorts of things. If you got that transversing through your blood, and you can get a closing of this, and that's not good. Once that closes up, you'll have a heart attack, or worse, you'll have a blood clot in the lung. It can be very, very serious, and so you want to try to lower the risk of cardiovascular disease. So exocytosis shuttles molecules out. It's not limited to large molecules. Vesicles first enclose the particle, then merge with the plasma membrane, and out it goes. And there's certain functions of exocytosis that all of us experience. I probably more than most is sweating, and that's of course moving out volumes of liquid. There's like 4 different types of ways that we sweat, and they differ by the degree or how much water they can move, but it is a process of exocytosis. So that's one way to look at that. And so the idea is it's sort of like endocytosis, but in reverse. It's kind of the same way. So to shuttle molecules out of the cell, particles within the cell are first enclosed in the vesicle. Then the vesicle moves through the cytoplasm to the plasma membrane, where the membrane of the vesicle merges with the plasma membrane, and the material in the vesicle is expelled from the cell. And so that shows you the universal sort of mixture of that phospholipid with membranes and the like. It's an ideal reusable bag, I guess. When you go to a grocery store, it would be like ripping your skin off, putting your food in it, and then putting it. No, that's not a good analogy. Forget that. So in and out of the cell is, of course, we're moving things. If you remember the FedEx delivery guy, and there's the shipping department, the Golgi apparatus, and we have various vesicles that the FedEx delivery guy has to deliver, and then that wherever that bag of goodies needs to go, the kinesian will take it there, and then it melds with the membrane, and then releases all of the goodies. It's sort of like Santa Claus bringing its gifts and merging with the membrane and releases them. I mean, how wonderful is that? And so the good old kinesian. So when materials cannot get into the cell via a diffusion or a pump, cells can engulf the materials and their plasma membranes, called endocytosis. And just in reverse, exocytosis. So in both processes, plasma membranes, the phospholipid bilayers, move to surround the molecule particles, and it pinches off. And the thing is, it's that universal sort of transport system that there's no waste. What an ideal system. It's just amazing how it can do that. Okay, we'll shift gears just briefly, and now we'll look at the genetic storehouse. We have genetic material in the nucleus. You can see here is the nucleus, and then we have 23 pairs of chromosomes. And if you were to break down each of the chromosomes, they're wrapped around these histones, the proteins forming a nucleosome, and that aids in the packing. Now, all of our DNA is packed in a certain way. We're going to see this in the lab. That makes a very characteristic size and shape of each chromosome. And so we have the 23 pair, and the pairs of course look identical to one another, but of that pair, let's say it's chromosome 3, it will look different than chromosome 4 due to the packing and the histone arrangement and the sequences and the like. So nothing new. We have the guanine and the cytosine, so the G's and C's and A's and T's binding, and that's your DNA. The nucleus is the largest, most prominent organelle in most eukaryotic cells. And see, I used to work in a business where we had patents, and I stored the patents in a safe. And that way, the original intellectual property of the company, which is where the value is, was safe in the safe. And so the nucleus is like that. The nucleus allows certain mechanisms to read it, but never to utilize it in a process. If you catch me, the messenger RNA is created. So that's sort of like I'm copying from the original and making a sort of a transcript, and then that messenger RNA is used to build. And so the original DNA is never really manipulated or altered in the sequence. It's preserving the importance of that information. And so it's kind of unusual to have these sugars, the guanine, cytosine, adenine, and thymine, actually convey information to do amazing things. And it's just very, very interesting. We're going to talk more about that when we get into biotechnology and the like. So the predominant structural components, we have the nuclear membrane. If you notice it, it's double. It's sort of like it's making sure that you're not going to drive a truck through there, a terrorist or something like that. It's really protecting it, and it has a very unique pore that only allows like messenger RNA and various things in and out of it. And so it's a very specialized structure to protect the DNA, but still allows the nucleolus to do all the timing and making sure the messenger RNA is producing things that we ultimately going to be needing in protein form. And so the nucleus regulates, controls, creates, does it all in terms of the intelligence of the operations. In other words, the instructions and all that. Pretty cool. And then the pore allows things to move in and out. And so what's moving in and out? Of course, the chemical information, which is sugars. All of those four bases, they're sugars. And so isn't that interesting that we convey information, very important information, in the form of these sugars? And then it's translated later, you're going to find out, with the transfer RNAs to make, now translating from a sugar to a amino acid or a protein to build a protein. There's no other example of that in life that I can tell you that sort of translation occurs. Very, very unique. So if a cell has the size of a large lecture hall or movie theater, the nucleus would be the size of an 18-wheel semi-tractor truck parked in the front rows. So that kind of shows you the size relationship. Your book chose that. It's probably not the best one I would have used, but nevertheless, you paid for the book, so I used the book. The nucleus has two primary functions, both related to the fact that most of the cell's DNA resides in the nucleus. First, the nucleus is the genetic control center, and the nucleus, being that, directs most of the cellular activities, controlling the molecules that are produced. Second, the nucleus is the storehouse of this information, and nothing really new there. So you can kind of see in the diagram that all of these smarts and information is stored here and packaged in a very smart and controlled way. Ultimately, replicating itself inside when the cells split, all of the DNA has to be copied. We're going to talk about that in later chapters, and I'll show you the fantastic enzyme that does this. There's actually, this is kind of misleading, it's a single enzyme that does both functions at the same time. So all I got to say is, wow, that's amazing. And it does it with 99.9% accuracy, which is again amazing. So the nucleus is usually the largest and most predominant organelle. It directs most cellular activities by controlling which molecules are produced and in what quantity. It is the storehouse, and three key structures: the pores in the nuclear membrane, the chromatin, and the nucleolus. And those are just reviews, and we talked about that. Cilia and flagella. Cilia are short projections that beat swiftly to move fluid along and past the cell. Flagella are long microtube-based structures that move cells through the environment. And you can see these, if you hear someone coughing, of course, we have that elevator of mucus, and smokers tend to damage the cilia, and you start to see the smoker's cough. Flagella, of course, using sperm cells that have these tails. In bacteria, we have flagella that spin just like that. So there's the cilia action, and it pumps and it actually moves debris and things out of your lungs. And cilia are amazing. It just they all kind of beat together in a pattern. Sperm cells, the flagellas actually spin, and one motion moves forward and the other motion kind of changes its direction. And birth control, by the way, prevents the sperm from moving around because it immobilizes the flagellum so it doesn't spin, so it kind of shuts down the motor there. So inner scaffolding of the cell, which is made from proteins of the cytoskeleton, it consists of protein fibers, microtubules, filaments, the same thing that sort of the kinesian was walking on, the cytoskeleton. So I just wanted you to remember that all of these things that we talked about with inside the cell and the importance of the nucleus, we have to have this inner scaffolding to protect the cell from compression and allow transport like highways. And even though the nucleus is the most important part of the cell, if we don't have the proper positioning, and everything has to work together, I guess is what I'm saying. It's just amazing, this concert of all these factors have to work together. So we'll stop here. I'll finish up with a little part of the section. I think it's 15 slides or so to finish it out. So that was part 4 I just covered, and I'll see you on part 5. And take care of yourself. And